Now we can look into the term MPF. MPF is maturation promotion factor. So it's a combination of both cyclin and CDK. So they join together can stimulate the mitotic and the meiotic phases of the cell cycle. This maturation promotion factor is having two subunits. The first one is catalytic subunit and the other one is regulatory subunit. Coming to the catalytic subunit, it can transfer phosphate from ATP to serine, threonine or tyrosine residues of a particular kind of protein. That means it can transfer phosphate from ATP to a particular kinds of protein. That means it's having kinases activity. See here in this combination cyclin and CDK. CDK is the catalytic subunit. CDK means cyclin dependent kinase. So this CDK is the catalytic subunit of maturation promotion factor. And coming to the regulatory subunit, that may be called as cyclin. So both regulatory subunit and the catalytic subunit join together to form this maturation promotion factor. Now we can go to the cell cycle checkpoints. There are mainly three checkpoints can be seen in a cell cycle. One may be at the end of G1 phase, another may be in the G2 phase, and the third one that can be seen at the middle of the M phase or mitotic phase. Now we can go into the details of cell cycle checkpoints. Before that, we have already studied cyclin E, cyclin A along with the CDK2 that drives the cell cycle from G1 to S, and the cyclin A, cyclin B along with the CDK1 that drives the cell cycle from G2 to M phase. Once the M phase got over, that means the daughter cells formed, then the cell cycle may be progressed or coming back to the normal. That means into the G1 phase. But this cannot be possible when the levels of cyclin A and B got high in the cell. So by that time, these cyclins get selectively distributed by different mechanisms in the cell. The same situation that also can be seen over here also. That means once the G1 phase is over, that means after S phase, the cyclin E, cyclin A, CDK2, these complexes got distracted by uh, different mechanisms. Generally, cell can progress from S to G2 phase. Those cyclins, along with the CDK, involved in the drive of the cell cycle from G1 to S phase are commonly called as G1 cyclins, and those cyclins and CDK involved in the drive from G2 to M phase are normally called as mitotic cyclins. Now we can go back again into the details of cell cycle checkpoints. Here you can see different phases of the cell cycle. They are G1, S phase, G2, and at the end you can see M phase over here. We have already discussed that there are three cell cycle checkpoints are there. The first one that may be at the end of G1, and the second one at the G2 phase, and the third and the last one can be seen at the middle of the M phase. Now we can go into the details of the first cell cycle checkpoint, that is at the end of the G1. We have already studied that the cyclin E, cyclin A along with CDK2 that drives the cell cycle from G1 to S phase. These cyclins and CDK2, they will be activated only at the end of the G1 phase. These cyclins and CDK2, they are normally called as G1 cyclins. See here, once the entire process that occurred in the G1 phase is over, then that activates the G1 cyclins. This G1 cyclin activates the CDC2. That CDC2 activates these pre-replication complexes to assemble on the origin of replication. Origin of replication means that may be seen in the DNA. So these pre-replication complexes gets assembled, then only DNA replication can commence. Once the DNA replication starts, once the DNA replication initiates, then we can say that the cell cycle that is driven into the S phase, because the major metabolism that is happening in the S phase is the DNA replication. So at the end of the G1 phase, these G1 cyclins activate CDC2, and that CDC2 activates the pre-replication complexes to assemble on the original replication of the DNA. Once it gets assembled on the original replication, then normally the DNA replication occurs and the cell cycle that may be driven into the S phase of the cell cycle. The second cell cycle checkpoint that can be seen in the G2 phase, we have already studied that the cyclin A, cyclin B along with the CDK1 that drives the cell cycle from G2 to M phase. That means once the entire metabolisms or processes that to be occurred in the G2 is over, then only these cyclins A, B and CDK1 gets activated, causes the cell cycle to be driven from G2 to M phase. That means these cyclins and CDK1 are activated only at the end of the G2 phase. These cyclins along with the CDK1, they may be called as mitotic cyclins. Mitotic cyclins. Here it is known as G1 cyclins. Here it may be known as mitotic cyclins. So see here, mitotic cyclins that activates CDC2. That CDC2 drives the cell cycle from G2 to M phase of the cell cycle. But the drive of the cell cycle from G2 to M phase is not at all simple, because the CDC2 over here is regulated. You know why? Because here in the G2 phase, the main thing that can be observed is the growth of the cell in volume. So as much as this G2 phase gets prolonged, the growth of the cell in volume occurs. So this CDC2 gets regulated by a kind of phosphorylation that is occurred in the tyrosine 15th and tyrosine 14th. So this kind of inhibitory phosphorylation that may be done by a protein that may be known as V1. So this V1 that phosphorylates CDC2 at its 15th and 14th residues. At the 15th residue we can see tyrosine, and at the 14th residue we can see threonine. So these two residues gets phosphorylated. This is a kind of inhibition. So this CDC2 is not at all gets activated by mitotic cyclins. Once the inhibition of this CDC2 by V1 protein occurs, then the progress of the cell cycle from G2 to M phase that gets blocked. That means the cell cycle that may be stuck in the G2 phase. So by that time, the growth of the cell occurs in volume. Once the growth is over, and another protein that may be known as CDC25 that removes the inhibitory phosphorylation of the CDC2. That means that removes the phosphorylation at tyrosine 15th and threonine 14th of the CDC2. That means that get writes the inhibitory phosphorylation to CDC2. Now the CDC2 gets activated, then the cell cycle drives from G2 to M phase. And one more thing that also happens at the end of the G2 phase, that is another protein that is known as POM1 that activates the CDR2, then that activates the CDR1 and NIM1 proteins that inhibits the V1 protein. Then the V1 protein is not at all functional to inhibit the CDC2. That also keeps the CDC2 in active mode. So the cell cycle that can drive from G2 to M phase. And there is one more checkpoint that can be seen at the middle of the M phase that we will discuss later during the metaphase of the cell cycle. So here we have discussed all the three checkpoints, one at the end of the G1, second at the G2, and the third one at the middle of the M phase that we will discuss later.